Good afternoon, everybody. I'm Jérémie Laurent, the founder and CEO of Astraveus. We are a company advancing next-generation platform to solve the bottlenecks of cell and gene therapy production. Our solutions are benchtop cell factories that are powered by microfluidics, and they aim at really revolutionizing the way process development and clinical manufacturing is done. So I will start with this little introduction of our company and team. We are a company about 10 years old. Over this period of time, we've built a team of 65 international talents. We are based in the south of Paris, which has been a great place to develop the company, raised about 55 million euros since inception. And we are making a lot of focus to be a patient and client-centric organization, and as well as building a strong culture that is focused on the development of people, and that is reflected by the values of the company you see on the bottom of the slide. So a glimpse into our labs, you will see that we are pushing limits of technology in Astraveus, and to do that, our recipe is to have key aspects of developing that technology under the same roof. So you can see on the left of the slide that we have developed expertise and assets in the development of microfluidic technology, as well as the related single use. I would argue that we are currently having the most advanced microfluidic technology, and we again fully own the manufacturing of the related consumable. We also develop systems and software around to turn this technology into products. And on the right side of the slide, you can see that part of our activity is also process development, performance evaluation, and that's because we don't want to just bring a product to customers, but also polished, optimized manufacturing processes that make the system really easy to adopt and use. So now to go a bit more concretely into what we're doing, and sorry for the PowerPoint issue we have here. On the right of the slide, you can see our solution. It's an automation platform that is a 6 feet wide machine. It has 10 production slots, meaning 10 production units that can run fully independently, so can run patient-specific material for potentially different types of products. Each of those units, so each of those production slots, is able to deliver a full end-to-end manufacturing process, starting from apheresis material, going until the fill and finished product. And additionally, each of those bioprocessing units is scalable. You can work at a downscale of a few million cells up to 1 billion cells per unit. So in that sense, it's not only an end-to-end manufacturing platform from the cell processing perspective, but also from the client and product journey perspective, from process development, potentially working with patient cells, up until large-scale commercial use. So the goal of this platform is really to multiply by 10 what existing team and facilities are able to do, because that low footprint system is delivering similar throughput as typically 20 or so people would do in 4,000 square feet of clean room. So the underlying technology behind that high throughput and low footprint platform is microfluidic bioprocessing. It's really a very advanced and novel technology. We are not using flasks or bioreactor, but rather, as you can see on the left of that slide, we are processing cells in microfluidic chambers exclusively. Those microfluidic chambers are similar to organ on a chips. They are a cell-centric, a biomimetic environment that is really thought to be the best processing environment for cells. And it's a very low volume cell processing technology, enabling to save at least a factor of 5 on the reagents. In our production unit that sits on the middle of this slide, we have a single-use element in blue, and this single-use element hosts a few hundreds of those microfluidic chambers that makes the unit able to produce up to 1 billion cells, and then 10 of those production units can be integrated in one production system. And that's how we have this factory on bench concept with 10 manufacturing slots embedded in a fully digitized, fully automated environment. Zooming a little bit into the technology now, this is really how the microfluidic chamber is working. It's a sandwich of pharmaceutical-grade plastic and gas-permeable membrane. The gas-permeable membrane is, of course, providing the gas exchange, which is contained in the lower half of the chip, supporting both hypoxic or normoxic processes. It is typically functionalized with biomolecules. And then the cells are bathed in 200 microns thick, approximately, layer of liquid, again providing a biomimetic environment which reduces the reagent use but also increases the concentration of cell-secreted compounds, which is favorable in many cases to the quality of cells. Then cell manufacturing processes typically start with cell selection, which is sometimes a bottleneck. We've developed a cell selection procedure which is integrated in those chips and is now a very reliable and high-performing process. So the fundamental principle is to coat this gas-permeable membrane within the microfluidic chambers with antibodies and play with the flow rate to selectively stick the cells of interest within the chambers. This technology has three distinct interests. The first one is you can start with raw materials such as apheresis. You don't need a prior step of purification. The second one is the cost of biotin-related antibodies is significantly cheaper, typically 100-fold, less than magnetic beads. And the last one is you don't use magnetic beads, so you don't have contamination whatsoever, and so you can have a simpler process without bead removal, and potentially also a shorter process. Then additional steps can be done. Here you have an illustration for CAR-T manufacturing. Still within those microfluidic chambers, you can perform activation, gene delivery, expansion, formulation, and harvest. Throughout all those steps, you keep the benefits of working within a very low liquid volume that is supportive of reducing the usage of reagents. Additionally, because the reagents are brought at very close distance to the cells, a few tens of microns, the contact is way more rapid and effective. Ultimately, it's diffusion, so random motion that brings the cells in contact with reagents. So the closer you are, the better this random motion is creating the contact between the cells and the reagents. Throughout all those steps, you also have embedded microscopy that provides a real-time cell count and visibility into how the cells are behaving. And last but not least, you can wash the cells and fill and finish the final container of the product. And actually, we do end-to-end, and beyond that, we cover part of the QC. We have taken advantage that we have embedded phase and fluorescence microscope in our system to explore if we could do basically the equivalent of flow cytometry, which we call image-based cytometry. The process consists in staining cells in distinct analytical chambers. Of course, they are separated from the manufacturing process. Then we take fluorescent images. We have a machine learning-based software that will segment the cells and quantify the signal at the individual cell level. And what we've been able to show is that very reproducibly, we have data that is in quantitative agreement with flow cytometry and tends to be more reproducible than a manual flow cytometry workflow. So to recap, the Lakhesys unit, the Lakhesys bioprocessor is truly an end-to-end bioprocessor within a very small format. It's really book size, a few inches wide and 1 foot long, and also enables to further streamline manufacturing by covering part of the QC. I'm now going to speak a little bit about performance evaluation data we've generated. We've announced beginning of this year the world's first microfluidic end-to-end CAR-T manufacturing. So this process was a very short process of 26 hours, 30 minutes microfluidic cell selection followed by 24-hour microfluidic activation, and last, a transduction of only 2 hours, which benefits from this microfluidic technology to be accelerated. The transduction was done at MY of just 1. As you can see, we have done a benchmark study with max and plate or max and G-Rex as comparison points. We have comparable. It's actually hard to increase beyond close to 100% purity or viability, but at least we are on par with the benchmark there. And on the transduction, you can see benefits of the microfluidic technology significantly enhancing the transduction rate, and that's something that we see reproducibly across donors. We then went on to explore and develop processes that are longer and incorporate expansion, still focused on CAR-T. So we've started by adding expansion to our process. We were able to show retaining purity and viability. In this video, you will see what users actually can see using the Lakhesys platform from the live microscopy feed. So the very small dots are the individual cells, which you can see are crawling over the surface, multiplying and then aggregating to form these clumps, which is normally what you expect. And of course, the nice part here is in a remote manner, you can really see if your process is on track and what the proliferation and the cell count looks like. So when this process was elaborated, we then went to add transduction, and this is the most recent data we've generated. So in this process, we have a 4 days expansion added to the selection, activation, transduction process. We are still seeing the nice purity, viability, and expression data. So overall with that, we are quite happy that, number one, we've been able to confirm the important metrics of cost reduction with our platform, and also to reach some important quality targets for ultimately CAR-T products. Of course, it's not the end of the game. We look forward to go much further in terms of process development, and we believe the advantages of the microfluidic technology combined with the process development capabilities of the platform will help ultimately to really bring the quality beyond what existing platforms are able to build. And so very much in that spirit, the stage that we are at now at Astraveus is to roll out our system more on the field. We have a first collaboration announced in which the product prototype at a full scale will be evaluated in third party, so that will be done by our partner NextGen in Leiden. They are a CDMO organization, and they will produce CAR-T with our platform and compare in head-to-head study with the current market-leading technologies. And I would say that we are generally interested to speak to all of you, whatever your profile is as manufacturers or developer of cell therapies. As I mentioned, we are focused on CAR-T, but already very interested in exploring other collaboration opportunities in other cell types. To wrap up this presentation, recap on our value proposition. We have a high throughput and low footprint manufacturing platform, ultimately enabling existing teams and facility, whether you are working with a CDMO or manufacturing in-house, to make 10 times more. The cost item is probably the most important. We heard it many times this week already, that it is really the main bottleneck of our industry. And because we are uniquely combining automation with a more efficient use of reagents with the microfluidic technology, we've shown through modeling that's been pressure tested with pharma ability to bring an order of magnitude change that probably is only achievable otherwise with in vivo or allogenic approaches. And last but not least, we believe our platform can help improving the time between prescription and infusion of the drugs. Of course, that's not something that Astraveus will do alone, but our technology supports rapid manufacturing of cell therapies and is amenable to decentralization. So I would thank you for your attention. I hope this was an interesting insight into what we are building at Astraveus. And please get in touch with us, whether through the conference or by email if you have an interest to discuss the technology and the opportunities to collaborate. Thank you.
Thank you, Astraveus.